Panie i panowie, dziękuję bardzo za zaproszenie. Well, to help Phil and others who don't understand Polish, I will now switch to English. So, happy birthday to the Institute. I was very, very impressed yesterday when we had a guided tour at the Institute, noting that you have had really very visible research in several neuropharmacology directions. Now with wonderful facilities, you can move to the next step. And I wish you a lot of luck for the future. So I visited Kraków first time in 1970 as the student. And I spent 3 months at the University of Jagiello. And I must admit that from that moment I fall in love with Kraków and with the local people. And it has been continued since today. So what I'm going to do this morning is that I will try to tell you about proteins which are called neurotrophic factors. And I try to convince you that these factors have a great potential also in treating several neurological diseases, in particular the neurodegenerative diseases. So I will first say a few words about the Parkinson's disease. I was helped by Dr. Velarsh, who yesterday gave a wonderful introduction to the subject. And then I will give you a brief overview about what we know already, how growth factors work in Parkinson's disease. I do that largely because there is a widespread misconception about the results and their interpretation. And then I will switch to a protein family we discovered, and then I will walk you through with some of the also unpublished results. So as you know, Parkinson's disease is progressive neurodegenerative movement disorder where dopamine neurons located in substantia nigra pars compacta projecting to the caudate putamen degenerate and die. And at the moment of the diagnosis, I think we have lost about 70% of the fibers and perhaps 50 or maybe even 60% of the cell bodies. Well, these data actually demonstrate that there is a dying back mechanism in the degeneration, meaning that the problem starts actually here. First we lose synapses, then the axons are degenerated, and then the cell bodies die. And the process in general is pretty slow. But what I would like to emphasize that in addition to the motor symptoms, there are also non-motor symptoms. And from the point of the quality of life of the patients, these are actually extremely important. These are, among others, constipation, posmia, depression, apathy, sleep disorders, and the current drugs for Parkinson's disease don't treat any of these. So if we look at and ask why dopamine neurons die, what is the reason? One of the reasons is that this is probably a neuronal population which makes the world record in branching. A single human nigral dopamine neurons can make up to 100,000 synaptic contacts, meaning that you have to transport the energy, proteins, to 100,000 destinations from one place. And of course, this makes the life very complicated. Cell has decided not to transport proteins, but to transport messenger RNAs. And the local synthesis of proteins in the synaptic area and also in the dendrite axons plays a crucial role. So we also produce large amounts of proteins which are required for the dopamine synthesis and metabolism. And these also are actually transported to these 100,000 destinations. So I just remind you that we have altogether about 1 million nigal dopamine neurons, which compared that we have 10 in the power of 11 neurons in general in our brain is a small figure. How 1 million neurons can control our locomotor activity? Well, because they make 10 in the power of 11 synaptic contacts. This is equal to the number of stars in the Milky Way galaxy. So, not a small thing anymore. Then, of course, there are Lewy bodies. Piotr yesterday explained quite well in details. And then these are neurons which have spontaneous activity. They fire with a frequency in 1 second. That is required to have the constant release of dopamine. So, saying all that, we also know that maybe from 5 to 10% of the patients have inherited or familial form of Parkinson's disease, and 90-95% have sporadic. We know these maybe 2 dozens of genes which cause inherited Parkinson's disease, and alpha-synuclein mutations are actually the first one described almost 30 years ago. Although we have worked with dopamine neurons for a long time, we have to admit that we know very little why and how dopamine neurons die. So how can we develop effective treatment if we don't know how they die? So I think this is a very big challenge what we face. So if we put it very simple, so the situation in the patient brain at the moment of diagnosis is more or less like that. We still have 50% of the neurons alive. They make synaptic contacts, but eventually they will die. And then we have maybe about 15% of the neurons which are still alive, are able to make dopamine, but they have lost the synaptic contact, so they are not functional anymore. And then we have maybe about 10% of the neurons which are still alive, but have lost the ability to produce dopamine. And then we of course have dead neurons. Dead is dead, there is nothing to do. But this population is actually the target of the new so-called disease-modifying therapy. We try to prevent the death of these neurons, we try to regenerate these neurons, and also de-differentiate these neurons and regenerate these axons. We were looking for the drugs which not only prevent the death, but also are able to regenerate. And the regeneration means forming new axons, stimulating axonal branching, and also enhancing the activity of these neurons. Where currently all the drugs what we have in the market treat symptoms. None of the drugs is slowing down or stopping the disease progression. And that is a target for the new drugs. Well, I have to say that because we have these 100,000 axonal branches which are filled with endoplasmatic reticulum. Endoplasmatic reticulum plays a crucial role in neurons. So it's a place for the folding and secretion of 2 types of proteins. All secretory proteins, all proteins which are destined to the membrane or which are moving to different organelles. They are all made on the rough ER. Then this is also the place for the post-translational modification, but extremely important also for the calcium homeostasis and lipid biosynthesis. So perturbations in protein folding, but also in the calcium homeostasis and lipid homeostasis cause ER stress. Accumulation of the misfolded proteins or rapid release of calcium from the ER stores to the cytoplasm triggers so-called unfolded protein response. So it can be divided into two. Initially, it's protective. It tries to help the cells to get rid of the unfolded proteins, such as alpha-synuclein in the Parkinson's disease, or equilibrate the calcium levels in the cytoplasm. But when it is chronic, then it triggers the whole machinery into the apoptosis and triggers cell death. So there are three major ER-located receptors which are key players in the ER stress. And these are the IRA1, which is a receptor serine-threonine kinase, which has also the ribonuclease domain in the cytoplasm. And then the other one is also a tyrosine serine-threonine kinase, PERK, and then is a less studied transmembrane protein called ATF6. IRA1 is actually evolutionarily conserved. You can find it also in yeasts. So what happens is that when proteins are aggregated, they bind to the major chaperone BIP, which is dissociated from these receptors, and these receptors become activated. They activate the ribonuclease, which will reduce the messenger RNA load in the cell. No sense to make more proteins if you have trouble to fold them. And if that is not enough, PERK is activated, which is in phosphorylating initiation factor 2, alpha subunit, and actually shuts off the protein synthesis. So if this continues to be chronic, both this pathway, actually through the JNK and NF-kappa B, but also this pathway will trigger apoptosis. So what we have here is that we have a balance between misfolded protein quality control in normal. There is a disbalance to misfolded protein in disease. And what we're trying to do is to get back the balance. So there are papers showing that indeed in the dopamine neurons of Parkinson's patients, both PERK becomes phosphorylated and also the PERK pathway becomes phosphorylated by initiation factor 2 alpha. So what would be the possibilities to tackle these problems? One of the possibilities is to use growth factors. So neurotrophic factors promote neuronal survival, control the number of neurons in the nervous system, regenerate axons, and protect them also from the toxins and injury. So nerve growth factor actually was the very first growth factor discovered by Rita Levi-Montalcini. This is actually a picture I took from Rita's original work. This is a chick sensory ganglia. If you cultivate it without growth factors, it will die in about 1 week. But if you add this growth factor NGF, you see that these ganglia are staying alive months, and you see also very strong stimulation of the axonal growth. So Rita, and at that time her PhD student Stanley Cohen got the Nobel Prize in Physiology or Medicine in 1986. I have put here also Victor Hamburger, because he predicted the existence of the neurotrophic factors, and actually the discovery was made when Rita and Stanley were working in his lab in WashU in St. Louis. So the young people in the audience probably notice another thing, that all these people lived a long life. So when you select the topic of research, remember that there is a very interesting correlation. I think I also look quite young. So, though the key growth factors which are tested in the animal models and also in the human clinical trials belong to the CDNF family. So there are 4 factors: the CDNF, nurturin, artemin, and persophin. We can't forget about these because these 2 mostly function in the periphery. But CDNF and nurturin function in the brain, but also in the periphery. So back in 1996, we actually were publishing 2 papers demonstrating that RET, the transmembrane receptor tyrosine kinase, is a receptor for CDNF. Later, we also discovered the co-receptors for nurturin and persophin. But how it works is that CDNF or nurturin binds to its specific co-receptors, and then this complex forms a ternary complex with RET, which becomes phosphorylated on the tyrosine residues. And these then eventually signal in the cells, leading to the survival and neural outgrowths. So I just want to emphasize that neurotrophic factors play a very crucial role also in maintaining neuronal networks. So neuron 1, which makes a synaptic contact with is neuron 2, is actually firing to neuron 2. And GDNF, BDNF, and other neurotrophic factors are synthesized and released in an activity-dependent manner, which means that if this neuron is not firing here, GDNF and BDNF are not secreted, and this synaptic contact is not maintained. But a new is formed between neuron 1 and neuron 2, which gets the electrical signal and eventually secretes neurotrophic factors. So these neural networks, as you know, are the basis for the memory and learning. So why GDNF is a drug candidate for neurodegenerative disease? Well, it can protect dopamine neurons. It stimulates growth and branching and regenerates axon. It can actually stimulate synaptic transmission. It can upregulate tyrosine hydroxylase. That's a key enzyme for the dopamine synthesis, but also increase the activity of the dopamine transporter. Dopamine transporter is actually quite important in regulating the trafficking of dopamine between the cell and the extracellular space. And then GDNF itself, it was published actually in the original paper in Science when GDNF was discovered, can stimulate uptake and release of dopamine. So from early on, actually, Barry Hoffer, at that time in Denver, was the first one to demonstrate that GDNF can protect and regenerate dopamine neurons in the mouse model of Parkinson's disease. The first in vivo data were published already in 1994. When indeed we can say that intracranially injected growth factors, because they don't pass the blood-brain barrier, you have to deliver them directly into the brain, that then they can protect and regenerate axons. But that is a widely spread misunderstanding. Usually people say, oh, GDNF works wonderfully in animal models, but it doesn't work in human trials. Not correct. Actually, GDNF works poorly or doesn't work at all in severe neurotoxin models. And patients who are treated, they have lost 80% of the dopamine neurons. If I take GDNF or nurturin and take a rat who has lost 80% of the dopamine neurons, nothing works. So there is no conflict between animal data and human data. It's just we have used different models. So there is one very interesting thing, and that is that there are several publications showing that GDNF has no effects when alpha-synuclein is overexpressed by the viral vectors. I think Piotr and also My lab has now, and several others have now provided data that this is actually an artifactual situation. If you overexpress alpha-synuclein, you really intoxicate the brain by the overexpression. It doesn't sort of reflect the real situation. I will show you some data on human dopamine neurons, which shows that GDNF actually works. So, of course, GDNF can be delivered as a protein or by gene therapy. By the way, there is actually recent, very exciting data from your countryman, Chris Vankiewicz, who using GDNF gene therapy has really, it's still unpublished, got extremely convincing data that GDNF works very well also in patients. So another sort of area of misunderstanding is a clinical trial. So let me explain that, how the situation really is. So there have been 4 phase 2 placebo-controlled clinical trials with GDNF. So if you summarize, no clear clinical benefit in 2 first trials, a clear effect on half of the patients in the 3rd trial, and then really very promising new data from Chris Vankiewicz in GDNF gene therapy. So what was the problem with the first 2 clinical trials conducted by Amgen? In the first one, the GDNF was injected into the ventricle. And as a result, because ventricle is far away from dopamine neurons, GDNF never reached dopamine neurons. So it's very clear. It did not reach putamen and substantia nigra, and of course, you can't get any response. They were very systematic. They started by injecting 25 micrograms to the patients, no effect. 100 micrograms, no effect. 250 micrograms, no effect. Eventually, 6 patients got 4 milligrams. It would be equal that you would drink 20 liters of whiskey. So, of course, you will be dead. In the second trial, again, the problem of the dose and delivery was a problem. So it was not GDNF what was failing, but it was the planning of the clinical trials. You can't blame the people because these were pioneering studies. We didn't know. I was actually in the advisory board of this first trial, and we didn't know how much to inject, where to inject, how often to inject. It was all open. Now, in the 3rd clinical trial conducted by Steve Gill and the Genesis, the Canadian company, they put 2 catheters into the putamen and then put a single outlet behind the air. So the patient came to the clinic, and from the external pump, GDNF was pumped to 4 places into the putamen. And it was done once a month, because GDNF, we know from animal experiments, has a very long-lasting effect. Well, I don't maybe need to go to the details, but in half of the patients, they saw a large clinically important motor improvement. And when they did the fluorodopa positron emission tomography, they noticed that all the patients who received GDNF, there was an increase in the signal, which might mean either that cells were protected or their activity was increased. And then, last but not least, but very, very important, it was well tolerated and absolutely no side effects were observed. So now I show you the video of some of the patients, how they look. So this is a guy before the GDNF, and this is after GDNF. And then the next patient, this guy can actually go to Olympics. And then you see from the 3rd person that it has not only effect on the global motor activity, but also for the fine motor activity, which I think is really, really important. And as I said, Chris Mankiewicz now has obtained in all patients, he had 8 patients, a very reasonable effect on CDNF gene therapy. So, but there are still problems with this CDNF. And the problems are that they don't pass the blood-brain barrier. That immediately raises a problem that ethical committees do not allow to treat early or mid-stage patients. When we asked the permission from the Helsinki ethical committee, we were not allowed to take patients earlier than 10 years after the diagnosis. And we did a PET for these patients. They had 85% of the dopamine neurons left. I actually thought that it was unethical to treat these patients because we knew in animals it won't work. But you have difficulties to complain with people in ethical committees, as you know. So intracranial delivery is risky, expensive. We calculated that a single patient in Helsinki University Hospital receiving this stereotactic injury, the cost was about 70,000 euros. It's really expensive. And then one of the major proteins, CDNF and nurturin, which are very basic proteins, is that they don't spread. So in the trial where I showed you that half of the patients had the effect, CDNFs covered 40% of the coded protein, which, if I translate it into the clear language, 60% of the dopamine neurons did not meet CDNF. So you can't rescue them. So we need to improve the spreading. And then, of course, none of the non-motor symptoms were treated because they do not spread to other brain areas in the human. So the question is, can we discover better factors? So we, quite many years ago, at that time my graduate student Päivi Lindholm and I discovered a new growth factor we called CDNF. We sent the paper to Nature, it was accepted, and then I got a phone call from the very angry lady from the International Union of Applied and Poor Chemistry. And she, without introducing herself, said that, Dr. Saarma, you can't use the word, I called it in the beginning, conserved dopamine neurotrophic factor, because it's evolution conserved. You can't use word conserved. I said, why? You just can't. Well, the older is older than I, so we had already the paper ready. I didn't want to change the abbreviation CDNF, so I invented cerebral dopamine neurotrophic factor. So, well, it turned out to be a very, very exciting and surprising factor for several reasons. First of all, it's largely intracellular protein located in the lumen of the endoplasmatic reticulum. And it is secreted because it has a signal sequence only in the injury and ER stress. It is evolutionarily conserved, and it functions when you have lesion or ER stress. So we solved the structure of the molecule. It turned out to be with a unique structure consisting of 2 domains, the N-terminal domain and the C-terminal domain. And we later found that actually C-terminal domain is a so-called business end. And it's very, very different from the conventional neurotrophic factor. GDNF, BDNF, they are dimers. CDNF is a monomer. And these are secreted and these are located inside the cells in the ER. Well, of course, we did the knockout. I very much hope that we will have a massive loss of nigrostriatal dopamine neurons, but eventually we found that there is an age-dependent loss of enteric neurons in the submucosal plexus. So actually the mice model the Parkinson's disease because they end up with a constipation. And then we also found not loss, but very significant functional changes in the nigral dopamine neurons. So we then started to search for the receptor, and of course, because locus operandi is ER, we were looking in the ER. Well, most of the work has been done by 4 very talented ladies, and I have circled a man demonstrating that I have some men also in my lab. So it started actually when Maria Lindahl, a postdoc at that time in the lab, did a month knockout and noticed that in the month knockout, the unfolded protein response pathways are fully activated, demonstrating genetically that it is really regulating the unfolded protein response. We did also the knockout in Drosophila, and we saw here also very strong activation, at least of the PERK pathway. And also in the CDNF knockout, we eventually saw the activation. We then used the specific inhibitors of PERK and IRA1, and PERK pathway, and demonstrated that we can block the neuroprotective activity of MANF and CDNF completely. Further telling that these UPR sensors might have something to do with the receptors. So the question we raised is CDNF protecting cells by directly interacting with UPR sensors? So this mostly work done by postdoc Vera and graduate student Olesia. So we use the microscale thermophoresis. You have the wonderful Dianthus instrument in your lab now, and demonstrated that CDNF with reasonably high affinity can bind to all 3 receptors. PERK having the strongest affinity. We replicated that also using the functional fluorescence complementation assay and demonstrated that this interaction occurs also in the cells. And then we mutated some key amino acids which we predicted by molecular modeling in the IRA1, the receptor, and demonstrated that indeed when we mutate these, we can block the CDNF effect. We then used mutagenesis and modeling and came and actually identified very clearly the areas where CDNF binds to the PERK in the structure. We did that by using chemical cross-linking, followed by mass spec and proteomics. So we identified the amino acid sequences which are involved in ligand receptor interaction. So to put more effort to, we generated a series of mutants, tested their binding to the receptors, and showed that K112 and also Y102 failed to bind to the receptor. And then when we looked at the signaling of these 4 pathways, we demonstrated that wild type CDNF regulates them, mutants don't. And then we tested them in mouse and human iPS-derived dopamine neurons, and we demonstrated that 3 mutants are completely unable to rescue most human and most neurons, and 2 of the mutants actually located in the cysteine loop in the C-terminal domain are actually fully active. So we have shown that both CDNF and MANF use MANF in particular IRA1 and CDNF PERK as a primary receptor. They reduce oligomerization of iron, they reduce phosphorylation, and also reduce downstream signaling. So we strongly believe that they are the major key attenuators of pathological chronic signaling of these receptors, and therefore have great potential also in neurodegenerative diseases. We then switched to test the CDNF effects on cells in animal models and then moved to clinical trials. So we first set up the dopamine neuron cultures from human iPS cells, and here we show that these neurons, actually they're largely developed by the postdoc in the lab, Li Yingyu, that they express the H, they express also dopamine transporter. In the higher mag, you can see also the expression of dopamine transporter and a little bit of TH also in the axons. And then when we treat these cells with 6-hydroxydopamine, we see a significant loss. And then CDNF can really rescue it. So we tested here actually the effect of CDNF on wild type human dopamine neurons. You see already effect at 10 nanomoles. But then we also tested the human dopamine neurons carrying the disease-causing mutation in alpha-synuclein. This is actually the first gene identified in American-Italian family in 1997 as a cause of familial Parkinson's disease. And you can see, we can rescue these quite nicely. We then, of course, were very interested, because that was a dilemma about GDNF. That GDNF is not working when alpha-synuclein is aggregated. So we actually tested GDNF on human dopamine neurons, also another postdoc was, and you can appreciate that actually already at 1 nanogram per ml, we can see the rescue of the cells. And roughly about 50% of the neurons are killed with a 6-hydroxydopamine. We then did the same assay with human dopamine neurons carrying the alpha-synuclein mutation, and you can again appreciate that we can rescue. But you notice that the number of cells we rescue compared to the wild-type cells, you see here 80%, here 60%. So that is a difference. So we all have also now looked whether GDNF can induce neurite outgrowths and axonal regeneration. And here we see a dramatic difference. Actually, in mutated cells, we see barely regeneration of the neurons. We then switched to rodent and then new non-human primate models of Parkinson's disease. These largely work of the former postdocs, Merja Voutilainen and Mikko Airavaara. Mikko is now the full professor of pharmacology, and she's an assistant professor of pharmacology. So she did the 6-hydroxydopamine models, and Mikko did MPTP models. And what we can show is that CDNF, as efficiently as GDNF, protects nigral dopamine neurons in rat model of Parkinson's disease, and also reverts the pathological ipsilateral rotation in the Ungerstedt turning model. We also then showed that using the alpha-synuclein preformed fibril models, the paper is published a few years ago in molecular therapy, that actually CDNF directly interacts with alpha-synuclein, inhibits its neural entry, and actually alleviates motor defects in mouse model of Parkinson's disease. But we were, of course, very interested to study the effect on non-human primates. And we, in collaboration with Judy Cameron at the University of Pittsburgh and with Don Gash's lab at the University of Kentucky, conducted a Michael J. Fox Foundation funded test of CDNF in these monkeys. So we had actually aged rhesus monkeys, pretty expensive. One monkey is about $15,000. We had 4 groups, MPTP, and MPTP with a low dose of CDNF, high dose of CDNF, and then positive control of GDNF. This was selected based on the Don Gash's work earlier published in Nature. And we did a unilateral lesion with MPTP, injecting it to the right carotid artery. And as you can see, we get a very nice unilateral lesion. About 70% of the dopamine neurons are lost, also shown in the post-mortem staining. And you can see also how well CDNF spreads in the monkey brain compared to GDNF. The spreading volume is about 4 times larger. We then looked at the UPDRS score. GDNF, as you can see in this rather strong lesion, has a trend but doesn't reach statistics. Higher dose has very little effect, but the optimal dose has very nice improvement. What is more is that we then also tested the fine motor control. We injected into the right carotid, so the left hand is paralyzed. And we measured how often monkey takes marshmallow or banana with the left hand, and compare that with the use of the right hand. And you can see that in control and in the GDNF, they didn't use the left hand at all. But here, actually, both doses, every second time they use right hand, they use also the left hand. So we have an improvement in the fine motor control. Well, we see also the rescue of dopamine neurons. We see the regeneration of axons, and we see also increase of the dopamine in the striatum. There is also a trend in the serotonin, but that's not statistically significant. So because CDNF is very well spreading in the brain, and it reaches the nearby ventral tegmental area, which is involved in the motivation and apathy, we were looking on the non-motor symptom motivation. And you can appreciate that with both doses of CDNF, we can actually significantly improve the motivation. GDNF has absolutely no effect. And here we see a robust increase of the dopamine levels in the nucleus accumbens, and also very strong upregulation of serotonin in the nucleus accumbens. The reason is very simple. When we measure by ELISA the levels of GDNF and CDNF in the nucleus accumbens, we have 100 times higher concentration of CDNF. It just spreads better. So we showed that CDNF protects and repairs dopamine neurons in rodent and human primate models. This is actually the first demonstration that a growth factor can treat non-motor symptoms. It can regulate the levels of dopamine, and the first demonstration of the regulation of serotonin by CDNF. And I think CDNF is better than GDNF in treating both global and motor symptoms, and notably better than in treating non-motor symptoms. And this was actually the first demonstration that CDNF and also GDNF can protect and repair human dopamine neurons, and human dopamine neurons carrying the alpha-synuclein mutation. Well, CDNF still, I'm about to finish in a few minutes. So there is still a big problem. You need a stereotactic surgery to get CDNF into the brain. So we were looking whether we can develop peptides which have the properties of CDNF but can pass through the blood-brain barrier. And we were lucky. When we purified, together with Päivi, the CDNF, we noticed that it is cleaved in between the two domains, the N-terminal domain and C-terminal domain. So, and then we see that also in low concentrations in the blood of human patients. So we then, that was actually work I did with my own hands. I'm very, very proud of that. So we tested the activity, and it turned out that this C-terminal domain is fully active. Then it was, of course, a question, does the C-terminal domain preserve the structure? And we did the high-resolution NMR. This is a full protein, and this is a C-terminal domain. They are virtually identical. I have superimposed them in the left corner. So we have a 61 amino acids long C-terminal domain. It protects motor neurons, mouse dopamine neurons, and also human dopamine neurons. And what is most exciting, when we tested it, we noticed that it enters the cells, it enters also the brain, passing through the blood-brain barrier, and then using different methods, we measure the fragment concentration in the brain. So we get maybe 2-3% of the systemically injected fragment into the brain. We then went to test it, and we see very nice improvement of the locomotor activity and also the protection of dopamine neurons when we inject it directly into the brain. When we do the systemic injection, we see a very nice improvement of the locomotor activity, but we don't see neuroprotection. So what is the reason? The reason is that the fragment is not sufficiently reaching the brain. The concentration what we measure of the fragment is about 1 nanomole, and 1 nanomole is too low. And the second reason is that it's pretty rapidly degraded. Half-life is about 60 minutes. So the fragment protects and regenerates mouse and human dopamine neurons in culture. It passes through the blood-brain barrier, can be used for systemic delivery. But for clinical trials, it should be optimized. The stability, perhaps also efficacy, but in particular blood-brain barrier penetration properties should be improved. So together with colleagues, Heikki Rauala and Eero Castrén, we started a company called Neuropharmacology, recruited our former postdocs to work at the company, and then tested first CDNF in phase 1, phase 2 clinical trials. And in this trial, the primary endpoint was met, and in some of the patients, we actually saw also some effects of neuroprotection. But because the very late stage patients, the effect was modest. But now we have actually licensed this CDNF fragment technology to this company, and they have now tested their own fragment, which has better pharmacokinetic properties, and they have already conducted successfully the phase 1A, and is currently conducting the phase 1B. So well, there is a hope that when we treat patients by systemic delivery, we can start the treatment immediately after the diagnosis because there is no surgery. And if we are successful, we can possibly also treat several of the non-mortal symptoms. Remember, CDNF knockouts develop constipation. So I will stop here and thank my colleagues and from my lab, and then people from Herantis, and then Mikko Airavaras and Andri Domanski's group, and Merja and her students. Thank you for your attention.
Thank you, Professor Saarma, for the fascinating story of CDNF and other neurotrophic factors. And we still have a couple of minutes for discussion, so I'd like to encourage you. Any questions?
We have 8 minutes.
Please, Professor Moreni.
Thank you for your very good lecture. And I would like to ask your opinion on the transplant of fetal stem cells that apparently work well in Parkinsonian patients. Do you think that at the basis of that effect could be the presence, the release of kinds of growth factors, could be the CDNF, BDNF, GDNF? What do you think about the role that these growth factors could have in the transplant?
Thank you for the question. So already in mid-80s, Anders Björklund's group in Lund conducted the grafts from embryonic brain into the Parkinson's patients. And in this pioneering study, he, together with Ulla Lindvall, demonstrated that in some patients, the effect was quite good, but by and large, this procedure was not successful. Now, when we have the possibility to develop stem cell derived either from induced pluripotent cells or from embryonic stem cells, human dopamine neurons, now 4 companies are conducting phase 1 studies on stem cell derived cellular grafts. It looks very nice. But if you look closer, there are several problems. And one of the problems is that, first of all, the survival of the grafted cells is very poor. You have to replace 500,000 of dopamine neurons because about half of the million neurons have died. So in theory, you should put there 1 million cells maybe. They put 50 million cells, because more than 10%, only 10% or less are surviving. That's the first big problem. The second big problem is that the grafts are not made to the nigra. They are made to the caudate putamen. If you graft to nigra, they all die. So if you graft to caudate putamen, you graft to the wrong place. They will make wrong connections because the cell bodies are located in the nigra. Well, the third problem is that especially with iPS cells, you have, well, although marginal, but still a risk to get tumors. So we will see. Now 4 labs, one in Lund, together with a Cambridge group, one in Kyoto by Dr. Takahashi, one in New York, and actually one in Australia, are conducting phase 1 clinical trials. We will see what are the results. Now these 4 companies or groups have very closely collaborated, exchanged data. I really think it will be successful. But it is not as fantastic as it looks at the first glimpse.
Mary, I had first, the lecture was a tour de force. I really loved it. I had a question about the CDNF data, which looks really good. So when you use 150 micrograms in your studies, you had a fabulous, very, very profound effect. When you went up to 450 micrograms, it looks like an inverted bell-shaped curve.
It's a bell-shaped curve, yes.
It falls off, but very quickly from a pharmacological standpoint. Is there any explanation for that?
Yes.
Okay.
So thank you for the question. So first of all, I did not want to use 450 micrograms. It was a Michael J. Fox Foundation panel who pressed me to use equal dose with GDNF. I tried to tell them that, look, we have rat data and we have shown that the optimal concentration for CDNF is about 10 times lower than that for the GDNF. But, well, they offered me $1.5 million. So I sold my soul. So that what happens is that, and that has beautifully shown with epidermal growth factor and also for the GDNF, and we have also some preliminary data on CDNF. So when you go at the high concentration of the ligand, then the cells are very smart. The GDNF binds to the receptor. Usually in the axonal termini, the receptor and the ligand is recycled, reused. They have this green technology for ages. But now it goes straight to the lysosome for degradation. And second, we have published a paper a couple of years ago in eScience where we discovered that there is a transmembrane phosphatase. And at high concentration, GDNF binds to this phosphatase and start to dephosphorylate red. So this is an internal regulation to get rid of the high toxic doses. So we see that also already in cells. It's a well-known, to find the right window is that way important.
So, it's incredible because it's very small in terms of molecular, it's very fine regulation. So the question is, if you go below 150 micrograms, you get an even more robust effect?
So we have done that not for monkeys, but we have done that for rats. We have started from probably 0.1 microgram, 1 microgram, 3 micrograms, 10 micrograms, 30 micrograms. So at 0.1 we see now nothing. At 1 we actually see behavioral effects, but no neuroprotection. At 3 we start to see also neuroprotection and beautiful behavioral effects. At 10 it's ideal, and at 30 it goes down. So at very low, no effect. At very high, almost no effect as well.
Thank you, Professor.
I also like to thank you for the astonishing lecture. 2 questions. As we are aging, we are losing not only dopamine neurons, but a couple of other neurons. Do you think that these therapies could be effective in, for instance, in dementia? And number 2 is, the other question is, don't you worry that this therapy could induce carcinogenesis? Thanks.
So there were 2 questions. One is that could these growth factors help also with aging and dementia? There is a beautiful series of work on the unregulated factor MANF, which is actually much more abundantly expressed in the body. And there is Dr. Jasper's group at the California Institute of Aging published a paper in Science a few years ago where they showed that the levels of MANF are declining in aging. And we have some experiments when we're overexpressed MANF in flies, they live longer. So if you want, you can buy MANF from me. But concerning, more seriously concerning the dementia, we have tested CDNF in mouse model of Alzheimer's disease. And we have shown a very clear effect in the improvement of memory. And as decent scientists, we have also controls. So we have also actually memory improving effect on naive animals. So quite interesting. But this is again brain delivery.
Professor Saarma, thank you for the fascinating lecture and very promising data. I would like to ask you about the observation that you show increase in serotonin signaling, considering the no effects of CDNF in dopamine survive, but increases in locomotor activity and motor improvement. Don't you think that this factor may change the phenotype of the serotonin neurons and they start producing dopamine, or and serotonin has a role in
Yes. Well, actually, my former postdoc, Jaanus Andresoo, who has developed the GDNF conditional knockout mice and also the GDNF hypermorphic mice, has shown also recently he had a paper in molecular psychiatry that also CDNF affects serotonergic neurons and has a robust effect on the serotonergic neurons in the rougher nucleus. With CDNF, we have only in vivo data, so I can't say much more than what I showed. So we have not yet tested the effect of CDNF on the serotonergic neurons in culture. We have not seen the effect. What I suspect is that CDNF has a vast effect on dopamine transporter. So it is one of the factors which, as you know, dopamine transporter is also largely located in the ER and Golgi, and not so much on the cell surface. So the regulation of dopamine transporter is trafficking from the ER and Golgi to the cell surface and back. And CDNF, we have shown, not published yet, that it enhances the surface location of dopamine transporter. I anticipate that that might be also the case with the serotonin transporters. But we don't have any data on that.
May I have
Sure.
a question? Is there any way to stimulate endogenous synthesis of CDNF?
Yes, ER stress or injury.
Oh, I see.
So,
That is,
but no chemical way. We have not seen any. Well, you can use ER stressors like tapsigagin, tunicamycin, D-teotriptol, but I don't recommend to use them.
But I meant some alternative way to, let's say, these analogs and some mimetics, CDN mimetics, if there is any way to stimulate.
We just filed a patent on small molecules which mimic the action of MANF and CDNF. And yes, there are chemical compounds which work, bind to the same receptors, compete with CDNF and MANF, and work also on animal models. Yes, that's the way we go now.
Thank you very much. Again, Professor Saarma, for this presentation. And now we have